Hey everybody, here we go with the video notes for Chapter 4, Cell Structure and Function. In this video, we're going to be looking at lots of different stuff. Cellular organization is going to be the major part of it, so think about how cells group together to form tissues and things like that. Looking at prokaryotic cells, which are the more quote-unquote basic forms of life, and then moving on to eukaryotic cells, which is pretty much anything other than bacteria, and looking at all the different organelles and systems within those that help maintain their lives, and then through those systems, obviously, our own lives. So a little bit of background. Cells, we began studying them early 1800s, really once we invented the microscope. That really obviously helped things a lot. And this is one of the unifying concepts in biology, really, evolution being the other one. And started with the work of two guys, Schleiden and Schwann. So this is Schleiden here, and this is Schwann. You can see operating independently of each other, but in the same general time. And the two of them together, or like through work that they started, and we built upon, formulated the cell theory, which kind of has three different parts. One, that all organisms are composed of cells, which, we take for granted nowadays, right? All cells come from pre-existing cells because they're self-replicating. Again, we take for granted, but for a while there was some kind of crazy beliefs about how cells sprang out of the air and magic and mystical forces and things like that. And then the cells are the basic unit of structure and function in an organism. So sort of like the monomer of a carbohydrate, like a big long carbohydrate like starch would be a glucose, same idea. The monomer, so to speak, of us are cells. And just think about how they're broken up, how they're divided. So you kind of see here's your plant and animal cells, and looking at differences between them, but also some kind of striking similarities, right? Like how these are like these columnar cells, and so are these, and like basal tissues underneath. But basically, cells group together. Similar cells will group together to form tissues, and then similar tissues can group together to form an organ, and then organs that do similar jobs can group together to form an organ system, right? So like all of your digestive organs, and then all of your organ systems then can work together to make an organism. So they work together. But you see how cells are the basic unit of life, the smallest unit of life. Sizes, just to sort of put things a little bit in perspective, is kind of hard on such a huge scale. But you have what we've been talking about so far, right? We had atoms way down here in the nanometer range. There's an amino acid, which makes up a protein. Viruses, and this is actually really a big virus. This is a bacteriophage. And then you start getting these are some organelles, right? The chloroplast is within plant cells. Bacteria range in size, and through here's your typical plant and animal cell, 10 micrometers to 100 micrometers, and then you've got some that can be bigger, like human eggs, frog eggs can get really big, and then you're talking like whole organisms and gigantic. And then obviously there's ranges in which you can see them, right? So you can see a human egg with the human eye, not really well, but you can see it. It's obviously better with a microscope, so all these things are visible with a light microscope, something we have in our classroom. And things that are really small, like these guys, are only going to be visible with electron microscopes, which are much, much greater magnification than a light microscope. So sizes, like we just said, ranging from 1 millimeter down to 1 micrometer. It's a mu micrometer. And the biggest thing that we talk about when we talk about cells is this surface area to volume ratio. This is a really, really useful concept. I know we've talked about it in other classes because it is so useful. You see it in nature all the time. And this concept of surface area to volume ratio. And basically, cells need this large surface area to exchange materials, to get nutrients in, to get oxygen in, but also to get waste out. And to do that, you need a lot of surface area to be able to get the stuff in. And the bigger you are, your volume is so great that it's hard to have a surface area that's big enough to get all these things through to you to maintain life. And so this is what really is what keeps cells so small. So a large cell, the surface area to volume ratio decreases which decreases the efficiency of transporting materials. So we say they have a small surface area to volume ratio. I'll show you some numbers in a second. And then small cells, so this is the preferred size by nature, they have a large surface area to volume ratio, which is more advantageous. So if you think about it like this, it's a lot of times it's easier just to kind of think of these square cells with numbers that are easy to see. If you've got this one big cell, it's 4 centimeters, right? Its surface area is 96 square centimeters, height times width times the number of sides. 96, and its volume is 64. And so it is a 1.5 to 1 surface area to volume ratio. Right? If you divide 96 by 64, you get 1.5. If we make it a little bit smaller, we see that the surface area is increasing, but our total volume, right, the total volume is the same, but we've just cut it up into smaller pieces. And so we have much, much more surface area. Now we're looking at a 3 to 1. And then if we keep going, it's a 6 to 1. So this is a large surface area to volume ratio. 6 divided by 1 is 6. 1.5 divided by 1 is 1.5. Right? This is a larger number. And so this is what you want to shoot for as a cell. You want to have the most possible surface area to exchange materials in and out as you can. So you're looking for a bigger number. Very important concept. We see this all the time, all throughout nature. This is why, like animals themselves aren't giant. This is one of the reasons, for example, our planet cooled down to be a rock as opposed to being a gas giant. It's all about the size and the surface area to volume ratio and the way it was able to cool. And so you see this surface area to volume all over the place, and you can apply it in lots of different places. So move on to prokaryotic cells. So again, these are our bacteria cells. These guys, their defining characteristic is we say they lack a membrane-bound nucleus. So they have no nucleus. The DNA is just kind of chilling out in there. Structurally, they're smaller and simpler than eukaryotic cells, which are the typical plant and animal cells. And we basically group them into 2 major domains. Bacteria, which you're probably familiar with. We tend to think of them as disease-causing things, and they are, but they're also crazy beneficial, right? They help us with a lot of food. Think about your yogurt and cheeses and stuff like that. Important decomposers, so returning nutrients and recycling nutrients within our own body, our own gut microbiome, which is helping us digest our food, useful making drugs and things like that. So we often think of bacteria negatively, but there's probably way more good things about bacteria than there are bad. And then there are archaea, which are similar. They're kind of like bacteria. There's some differences in their cell membrane and there are differences in their cell wall. And these guys live in extreme habitats. They're thought to be older, like some of the oldest forms of life on Earth, and so they live in crazy hot or crazy cold, crazy acidic, really salty environments, things like that. So structurally very similar, but they're biochemically different, which makes us think that they evolved differently. So again, prokaryotes really small, 3 basic shapes: sphere, rod, and spiral. And so you can see these are the coccus, bacillus, and spirillum. And so you see this sometimes a lot in their names. Coccolithophore is something you see in the ocean. They're kind of almost like a stony covering. That's what the lith comes from. Anyway, I digress. Those are just the same, the general basic shapes. So they all have a cell envelope. So this is familiar to us. This is just the plasma membrane. So this is the lipid bilayer that we've been looking at, those lipids. They have some proteins embedded in there. They can form what we call internal pouches or mesosomes. And so it's basically like an invagination or like a pocket. So that can increase the surface area. They have a cell wall. So like plants, they have a cell wall. It is still strengthened by peptidoglycan, which is a carbohydrate, so different. It's not cellulose. And then they have the glycocalyx, which is another layer of polysaccharides in the outside of the cell. So this is helping provide them with protection. So just looking at them diagrammatically plus a picture, you can see the different parts. So their DNA is just sort of free floating in there. They've got the plasma membrane, they've got the cell wall, they've got the capsule that's enclosing them, flagellum for movement. The ribosomes, we'll get more into ribosomes, but these guys have ribosomes just floating around, free floating in there. And these bristles, these fimbriae that help them stick to surfaces. So within them, you've got the cytoplasm, which you're probably familiar with, this fluid solution. When you talk about cytoplasm, it's the fluid plus all the stuff. So it's like the fluid, the water, the organic and inorganic molecules, the enzymes, things like that, all the extra stuff in there. If you wanted to talk about literally just the liquid, we would call that the cytosol. They tend to get used interchangeably, somewhat incorrectly. Usually you're probably referring to cytoplasm because it's all the stuff that's in it as well. The nucleoid, right, they don't have a nucleus, but there's usually a region where the DNA tends to kind of congregate. The plasmids are another type of circular DNA, rings of DNA. These are really beneficial in research because you can manipulate these plasmids really easily and kind of give bacteria different traits like being able to glow in the dark and things like that. And then our ribosomes, which you guys know about, but this is where proteins are synthesized. Externally, flagella, like we just talked about, the fimbriae to stick to surfaces, and the conjugation pili. So basically it's a tube that they can connect with each other and pass DNA, so basically having sexual reproduction. Moving on to eukaryotes. So these guys are going to contain a lot of the same, some of the same things. They have the membrane-bound nucleus now, so we have the special region for the DNA, and then specialized organelles, which we're going to totally get into. And then they have a plasma membrane as well, so this is separating them from the environment, and this is going to be helping to maintain the homeostasis of the cell. Homeostasis meaning same state, like keeping things regulated. So this is what's going to control things that move in and out of the cell. And this is, of course, that phospholipid bilayer that we've been talking about. And then we've got embedded proteins as well as some other molecules within the cell membrane that help the cell carry out its daily functions. The first two are what distinguish a prokaryote from a eukaryote, having a membrane-bound nucleus and then these specialized organelles. We tend to say membrane-bound organelles as well because you've got the ribosomes, which are not made of membranes, right? They're just made of ribosomal RNA, which we'll talk about a little later. So those are not made of membranes versus all the other organelles that you guys are probably familiar with, you know, the ER, the Golgi, mitochondria, those all are made of membranes. Those are something that only eukaryotes have, prokaryotes do not. They're much larger, and then they're compartmentalized. So all these organelles, the point of the organelles is to have specialized areas where the cell can compartmentalize different chemical reactions and give them the environment that they need for those reactions to happen as efficiently as possible. So they can isolate reactions from other reactions, like if they, enzymes we talked about needing special environments, like if an enzyme needs a certain trait, like a slightly different pH or something, right? You can create this little zone, this happy zone for that enzyme, so to speak. And so ways for the cell to efficiently carry out all the chemical reactions that it needs to do to maintain life. So just kind of some cool stuff. Looking at this is like an evolutionary tree, and it's kind of interesting because if you look, archaea is actually like split off the branch here that's giving rise to eukaryotes. You'd almost think that they would be along the same branch as the bacteria because they're so similar, but there's a lot of data that actually places them more here, and so more like that. This suggests that maybe the eukaryotic cells gave rise more from archaea than they might have from bacterial cells. Like this is the common ancestor between archaea and eukaryotes being right here versus a common ancestor of all life being right here. Kind of interesting. It's just expected to be here. This is a little bit more of a somewhat new discovery, so to speak. So how do we get eukaryotic cells from these prokaryotic cells? So kind of a couple processes. First, we think that the nucleus evolved from invagination. So we talked about those mesosomes, little pockets that could form. And so it's sort of envisioned that maybe like a mesosome formed and then somehow like totally pinched off and became like a bubble within the cell. And then the DNA, if it moves into that, like, oh, wow, this works really well by keeping our DNA separate. Allows us to do more. And then maybe similarly they could have gained some of those other organelles like the ER and the Golgi, so some like again invagination or making more pockets inside, allowing that to happen. And another cool thing is a lot of evidence suggests that mitochondria were actually their own aerobic bacteria that were basically like eaten by the cell or like engulfed by the cell. And then they sort of got this like symbiotic relationship where the cell's like, here, have some sugar. And the bacteria's like, thank you, I think I'll make some ATP. Would you like some of that ATP in return? Same thing with chloroplast. They think that was from a photosynthetic bacterium. And so eventually became part of the cell. We call that the endosymbiont theory, like symbiosis in an endo meaning within, so symbiosis within. So pretty cool. So yeah, explains the Golgi and the endoplasmic reticulum, this invagination of the membrane, and then like I talked about, the other guys were probably engulfed by a primitive eukaryote at some point, and then gave rise to all the other eukaryotes. So two main groups of organelles that we're going to talk about: the endomembrane organelles and then the energy-related organelles. The endomembrane are these guys that communicate with one another via these channels and vesicles, and so this is what's like moving stuff around the cell and sending messages forth and things like that. And then your energy-related, of course, are the mitochondria and the chloroplasts. These guys do their own thing. They're independent, self-sufficient. Oh, and I did forget to mention another bit of evidence in support of the endosymbiont is they also have their own DNA. So mitochondria have their own DNA. You may have heard that in genetics work. A lot of times they look at mitochondrial DNA because that is passed from mother to mother, mother to daughter, or mother to both children, because in a developing egg, all the mitochondria are moms. And so it's an interesting genetic lineage thing that you can look at. But anyway, independent and self-sufficient. So again, it's kind of showing us that they could probably, showing us that they probably came from something else. Mostly just put this in here for your own benefit in your note packet, or a handy slide for you to look at, a spot where you can see all the animal cells and kind of, or all the animal organelles, what they look like, a little bit of their functions. So just a nice resource for you. This might also be something, even if you don't print out the whole note packet, you could always go in and get that note packet and like print off this slide if you wanted to. And then same idea with the plant cell. And so a couple things to think about. Again, these pictures are just generalized, right? Like not every animal cell looks like this. Not every plant cell looks like this. Some have more of some organelles. Some have like none of other organelles. This is just generalized. So a specialized cell, like I said, could have different different complement of organelles. For example, liver, which probably know is the detoxifying organ, that has lots and lots of smooth endoplasmic reticulum, because that is what's producing those lipids that help detoxify. So again, these guys are all working together to help keep this cell alive. So just like our organ systems, our respiratory, our digestive, cardiovascular are working to keep us alive, these different organelles are working together to keep the cell alive. And so different molecules are being manufactured in different places, different spots for different uses. Sometimes they're shared with each other, they're moving back and forth, things like that. They get transported around the cell by these things called vesicles. So these are basically just membrane sacs, little bubbles of membrane. So you kind of like encapsulate it in that, and then you can send it from organelle to organelle. They move around using what's called the cytoskeleton. So skeletal, skeleton. Cyto is the prefix for cell. So these are protein fibers that basically, like it says, like railroad tracks, that they can move along. They help provide structure. We'll talk about those later on in the video as well. And then we also have the cell walls. We talked about these before, having cellulose as opposed to the peptidoglycan in the bacterial cells, to provide mostly structure. A lot of people think, oh, wall, protection. And yeah, I'm sure it does provide some protection, but really still it's the plasma membrane that's regulating stuff. The cell wall is really more for structure. So the nucleus, as you probably are aware, often called the command center of the cell, usually found near the center, but not always. It's surrounded by the nuclear envelope, which is, or nuclear membrane sometimes it's called, basically the same thing. It's a phospholipid bilayer. It has these pores in it called nuclear pores, which allows exchange between the nucleoplasm. So instead of cytoplasm, it's nucleoplasm. And so stuff can go in and out through those nuclear pores. They contain chromatin, which is in that nucleoplasm. Chromatin is basically is the DNA all stretched out. A lot of times we think of this as the chromosome form, so it can condense and make those little X-shaped structures that you maybe are familiar with from talking about like cellular division. Those are chromosomes. This chromatin that's condensed around proteins. There's also this dark area. It looks like the nucleus of the nucleus called the nucleolus. This is not where all the DNA is. Sometimes people think, oh, well, if it's the nucleus of the nucleus, that must be the DNA. Nope. The DNA is out here. This is where ribosomes are made in the nucleolus. Ribosomes, speaking of which, are composed of rRNA, so ribosomal RNA. So they consist of two units, a large and a small subunits. They're made in the nucleolus. This is kind of what they look like. They call this a simplified version. You got the large unit connected with a small. This is a little bit more what it might look like in 3D. And this is kind of trying to envision what it looks like all wrapped up and coiled. They can be on the endoplasmic reticulum, the rough ER, which we'll talk about a little bit, or they can be free-floating in the cytoplasm. They can be by themselves, or we can group them together in polyribosomes. And of course, the really important thing is this is where protein is made. And so this is where the magic of the cell happens, so to speak. How does this work? Is through those processes of transcription and translation, which, again, you've probably heard about in previous classes. DNA has the information to make a protein, but the problem is it can't leave the nucleus. It's too big to leave through those nuclear pores. Also, it's pretty important, so we want to try to protect it. So what we do is we copy or we transcribe that information onto a complementary strand of mRNA, which stands for messenger. That can leave through the nuclear pore. That goes out into the cytoplasm, hooks up with a ribosome. The ribosome reads that message and assembles chains of amino acids to make a protein. And so this is how we synthesize a protein. So this is transcription. This process is called translation. So we translate the message of RNA into protein. And so we call this the central dogma of molecular biology, DNA to RNA to protein sequence. And so here's just a diagram of that. Also, it could be helpful for your notes. You might want to consider printing that out. You got some translation going on here, DNA unwinding, joining up with the RNA, being read by the ribosomes, and then the protein can go off and do its thing. You know, maybe it's going to go into the endoplasmic reticulum and get modified and shipped off somewhere else. Maybe it's just going to be free floating in the cytoplasm and for that own individual cell to use and do something fun with. The endomembrane system, so endo meaning inside, so the inside membrane system. This is how we transport stuff around the cells, so in a series of intracellular membranes, so within the cell. Again, helping to keep enzymatic reactions in a certain spot, so you're not dissolving things that shouldn't be dissolved and reactions aren't happening where they're not supposed to be happening. We've got the nuclear envelope, we've got the endoplasmic reticulum, the Golgi apparatus, and then vesicles. So the ER, endoplasmic reticulum, these flat sacs, basically, they're continuous with the nucleus, and so they do kind of look similar to the Golgi. If you're ever unsure if I'm looking, hey, am I looking at Golgi or ER? ER is always with the nucleus. They can have ribosomes on them like this, which if that's the case, we call that rough ER. And so in this case, those ribosomes make the proteins and then immediately put them right inside the ER. The ER is going to modify them and process them. Maybe they're adding some sugars to them to get glycoproteins, packaging them up for export, things like that. And then they package them into these vesicles, and then they go to the Golgi apparatus for some more Golgi magic. The smooth ER doesn't have the ribosomes. This is where lipids are synthesized. So you could be making hormones produced there, or just making more membranes to help replace the membranes in the phospholipid bilayer. And like we said before, in the liver, this is also detoxification processes. And so liver has a lot of smooth ER because it does a lot of detoxification. And these also form transport vesicles to send stuff to the Golgi. So the Golgi, named for Camillo Golgi, an Italian scientist, Golgi with a soft G, not Golgi. You have these flattened curved sacs. So they do, you can see they look pretty similar to the endoplasmic reticulum. Again, this is going to be separate from the nucleus, right? They're usually closer. They're always, in pictures, they're always depicted closer to the cell membrane. Sometimes they say like a stack of hollow pancakes. I've heard people call them like stacks of grocery bags, things like that. Their main job is to process and package proteins for export. And so they get these signal sequences. So they're going to receive a protein, right? So remember, the protein gets made by the ribosome, gets put into the ER, maybe gets some synthesis, some carbohydrates added to it, puts it into a vacuole vesicle, sends it out. It joins up with the cis face, the inner side of the membrane, and as it passes through all these different stacks, it gets modified. And so it could be putting different proteins together, adding some other amino acids, adding other carbohydrates, doing whatever it needs to do to make that a more functional protein. Maybe it's refolding, and then it leaves through the trans phase. I like to think of trans meaning it's going to be transported away in these vesicles again, which then go and they can merge with the cell membrane and boop, go out and do their thing. So out into, maybe this is making lactase and it's being excreted into the lumen of the digestive system so that you can eat your ice cream and not get gas. Sometimes they go back to the ER to be recycled. Sometimes they can merge with another Golgi, something like that. But these could basically all get recycled. Moving on, lysosomes, another membrane-bound vesicle. They don't have these in plants. They're produced by the Golgi. And these guys have powerful digestive enzymes. I like to think of, lyse, right? Cells breaking. Lysosomes, these are going to help digest and break down cells and things like that. So they are for digesting larger molecules into smaller subunits or recycling cellular resources. So for example, if a cell were to engulf a food item, let's say, the lysosomes might help digest that and break it down, break a carbohydrate down into simpler glucose. This picture here, we've got an old mitochondrion, that's the singular for mitochondria, being digested because it's no longer functional, so they're going to recycle it. Maybe if you were like a white blood cell and trying to destroy enemy invaders, you might engulf a bacterium and then can send these lysosomes in there just to dissolve it away. So kind of like the cleanup crew of the cell. So yeah, along those lines, so talk about pathogens. This can also be the source of diseases. Common, or maybe, I don't want to say common, but a well-known example is Tay-Sachs disease, where it doesn't produce the correct enzyme, and so you can't recycle stuff the way you should. And kind of cool, nowadays we can, through gene therapy, can help restore the gene that produces these enzymes so that we can produce functioning lysosomes in these individuals. So, quick summary of our endomembrane system. Proteins made in the rough ER, lipids in the smooth ER, and then they're transported to the Golgi. The Golgi modifies these, packages them into vesicles, and sends them out to the cell, maybe other places in the cell, maybe extracellularly, through the vesicles. Exocytosis is things moving out, exiting the cell. Endocytosis is coming in. Example, mammary glands producing milk and pancreas producing digestive enzymes getting exported outside those cells. And then the lysosomes can fuse with incoming vesicles and digest macromolecules, or they could fuse with things that are already in the cell, like you said, like digesting broken down and non-functioning organelles. And then here's an overall picture of everything, showing what they do, kind of where they're located. Nice little resource for you. Some other organelles, microbodies, contain specialized enzymes and perform special metabolic functions. They are found in plants, animals, and some protozoa. They are not. They're similar to lysosomes. They're not lysosomes though. Lysosomes originate in the Golgi. These guys originate in the endoplasmic reticulum, so slightly different organelle. A common example of these is a peroxisome, again containing enzymes. And with a name like peroxisome, can you have any guesses as to what it might digest? If you said peroxide, you would be correct, right? This is going to help catalyze that reaction. to produce hydrogen peroxide. Oh, sorry, catalyze reactions that produce the hydrogen peroxide, and we can break that down later on with catalase. Vacuoles are just kind of like the vesicles, same idea, but they're bigger, and theirs are more for storage than for transport. Usually water plus other things. The central vacuole is the most popular example. This is in plants. It holds a ton of water, and you can see it can hold up to 90% of the volume, so it can really squish those other organelles out to the outside of the cell. For a couple reasons. One, you want to store a lot of water if you're a plant, but this is also good too because it squishes these chloroplasts out to the outside, so they're kind of closer to the sun, the incoming sunlight, so that's another advantage there. So mostly storage of water, but also storage of nutrients, some digestive enzymes in there too, so they can do some of the jobs that a lysosome might do. The development of turgor pressure, helping keep this cell full and rigid as opposed to like floppy. Think of like celery or carrot that's been in your refrigerator for way too long and doesn't have that snap anymore. That's because the vacuoles are empty and it no longer has what we call turgor pressure. They can use it for protection using toxic substances, and like I said, for digesting some things much like lysosomes would have. So moving on to energy-producing organelles. These are our chloroplasts and our mitochondria. The mitochondria, as everybody knows in the whole history of the world, mitochondria are the powerhouse of the cell. There was a meeting of biology teachers, and we all decided that we're all going to call them the powerhouse of the cell. These guys make ATP from carbohydrates. And then chloroplasts use carbohydrates to, or use the byproducts of mitochondria, the carbon dioxide in the water, to produce carbohydrates. And so you have this nice cycle where one benefits the other. So the chloroplasts, these guys have a double membrane. The inner membrane is folded a lot to form these disc-like thylakoids, which are stacked to form grana. So if we're looking at this over here, right, this is these are the grana, and then the thylakoid is each individual one, right? So this is a thylakoid here, and then this whole thing together is a grana. Lots and lots of membranes because this is, again, increasing that surface area, and this is where a lot of these reactions are taking place. We'll get into the nitty-gritty of photosynthesis later, but those reactions are taking place within those, and so by doing this structure, we get more cell membrane in which to have that happen. Green because of chlorophyll, that's the green photosynthetic pigment. It's only found in the inner membranes. There are other pigments too, but it's just mostly green, so that's why they look green. So again, they're membranous. Yes, we sometimes call them plastids. And as you probably know, they capture light to do photosynthesis. So they synthesize carbohydrates from carbon dioxide and energy using the energy from light. So solar energy, carbon dioxide plus water to make carbohydrates and oxygen. And this here is a little diagram of where these reactions, some reactions are happening in the cytoplasm, some reactions are happening within these grana. And again, nitty gritty, we'll talk about that later. So some more specifics. The chlorophyll is in the thylakoid. Enzymes are in the fluid, the actual stroma. That's this fluid here. So it's like the cytosol of the chloroplast, capable of conducting photosynthesis. And again, the endosymbiotic theory, we think that some photosynthetic bacteria was basically eaten by a proto-eukaryote and led to this construction here. Mitochondria contained in pretty much all eukaryotic cells. Not all of them do, like red blood cells don't have any don't have any mitochondria. They're smaller than chloroplasts. Numbers vary. So like if you're talking about a muscle cell, they're going to have lots and lots of mitochondria, right? Because your muscles need lots of energy. They do have their own ribosomes, and like I said before, they have their own DNA. They also have a double membrane system. The inner membrane surrounds the matrix and is very folded, convoluted to form what we call cristae. So all these little folds are called cristae. Again, additional surface area for chemical reactions. Way to boost that surface area to volume ratio. And then we talk about the matrix is the inner fluid substance that has some of the respiratory enzymes. So that's where they're going to break down the carbohydrates and then send those into the inner membranes to turn it into ATP. And so pretty much all the ATP we use comes from these guys, which is why we call it the powerhouse of the cell. And I'm sure you will remember ATP is adenosine triphosphate. That's the major energy molecule of the cell. All right, our last section here, the cytoskeleton. Lots of different functions, maintaining the cell shape, helping with movement, and then movement of the actual cell, if it's a cell that can move, but then also transporting stuff, movement within the cell. So 3 main types of fibers. We have actin filaments, intermediate filaments, and then microtubules. So these guys can be made and unmade as needed. Sometimes they're even like being made and unmade like as you go. It's almost like trying to move something heavy along the ground with logs, right? As soon as one, or it's like playing our half pipe game that we play before. Like as soon as the ball goes through one pipe, you can bring that down and catch it on the other end. What did they call that game that we play? Blue White Day. Anyway, so they can just reassemble on the leading edge. Kind of like the bones and muscles of animals. And again, dynamic in response to these environmental changes, gets made and unmade as needed. So actin filaments, extremely thin, like twisted pearl necklaces. So just like it sounds, like it's like a string of pearls that are twisted around each other. They can form a dense web just underneath the plasma membrane to help maintain that shape. So that's kind of what you're seeing here. This web, keeping it, giving its form. They can support the microvilli. So when we're talking about intestines later, they've got lots of little projections on the outside to, again, increase surface area. I told you it was important. So those help support those. Again, moving stuff around in the cell. If you were like an amoeba, those are those like blob organisms that can move around. They call that cytoplasmic streaming. They have pseudopods, fake legs that like this sort of like squish out from one side and then surround something. That's accomplished with the actin filaments. And then really important for muscles. This is our major structure in a muscle. You have two main filaments, actin and myosin. And so here's that string of pearls arrangement, a long chain that are strung around each other. And so here's your actin and muscles, and we'll get into that later too. Intermediate filaments, as you guessed, are intermediate in size, rope-like, and they do vary in nature from tissue to tissue. They do lots of different jobs, supporting the nuclear envelope. Cell-cell junction is a big thing, holding cells together really, really tightly, giving strength. And they kind of, you see they have this construction where you have a monomer. 2 monomers come to twist together to form a dimer. 2 dimers come together to form a tetramer, right? And then you put them all together, you can form this sheet, and then that sheet can coil to form this filament bundle. And so you can see how this is providing strength in holding things together tightly, like in our cell junction. And then the last, microtubules, hollow cylinders made of alpha and beta tubulin. And so these guys just like to pair together in dimers. So there's a dimer right there. And then they arrange themselves into these spirals of 13 dimers around. And so they just kind of go around and making this tube. So these are controlled by the microtubule organization center. The most important or famous one of these is the centrosome. You've probably seen this before, heard about it, like the centromeres. Interacts with the motor molecules kinesin and dynin to move organelles. I'll show you a picture of that in a second. And then this is also what's helping move chromosomes around. When we're talking about mitosis and moving chromosomes to the middle and then back away, we're going to be looking at these microtubules that are going to be responsible for that. And so this is that dynin and kinesin motor. So it's kind of cool. It just uses ATP to sort of walk along the microtubules. This is your microtubule. It's like the train track. And so this might be bound to whatever it is that's trying to move. And it's kind of walk along here. And then as this gets passed by, these can reassemble on this side to make new road or new train track for this guy. So there's all those guys together, showing a little bit about what they look like and different uses for them. Centrioles, we just talked about. These are short hollow cylinders made of the microtubules into 9 overlapping triplets. So these are 3s. These are like the anchors for mitotic division. And so like when those microtubules are anchored to here and pulling the chromosomes away from each other. These guys also are sort of like the basis for the cilia and flagella, which help bacteria and some eukaryotes move. So these are like the hair-like projections. So cilia are the many small hair-like projections, kind of like oars, versus flagella is one long whip-like. They are different from a prokaryotic flagella. They're structurally different. They look the same. They function the same, but structurally they're very different on the inside. They do have an outer covering with membrane, and then you have these 18 microtubules arranged in 9 pairs, and then 2 in the middle. So we call this a 9+2 pattern that's used by the cilia and flagella. So cilia, again, much shorter than flagella, moving like oars, like waving something along. So sometimes it's used for movement, sometimes it's used to move things. So like the cells lining the upper respiratory tract can help move dust back out or things back down or whatever. And then flagella moving an actual cell. Sometimes it pushes, sometimes it pulls. So here's this 9+2 arrangement, right? Looks pretty familiar. That's the basal body of the microtubule that we talked about, and then this is what it looks like inside the flagella. You've got 9+2. And that is the end.